here is how he tried to improve that technology. Instead of printing on a glass line or a membrane, he thinks we should just print on SDS patch gel, because only SDS patch gel can be used to do electrophoresis. Okay? And so for the first proof of concept, we try to stimulate A431 skin cancer cell with the EGF 10,000-fold higher than most other cells. That means if we give it a very little amount of EGF, there will be a lot of singlet. So it's easy for us to detect the singlet. So for the first time, we want to try some easier system. So we serum stamp the A431 skin cancer cell for 48 hours, and then we stimulate them with different amount of EGF for 6 time points, from 0, 1, 5, 15, 30 to 60 minutes. Because most of the signaling will change within an hour. And then we use an array, a Gaussian nanoproduct array, to print onto the homemade SDS patch gel. The array is a non-contact array. That means it just spit out the lysate without contact the gel. A non-contact array is very nice because it don't contact your gel, so you don't have a risk to get any pollution or anything else. So the gel is about the size of 96 well plate, and we print the same sample. That means 1 protein marker and then 6 samples from each of these time points onto a single well, and then we repeat that 96 times. That means in each well there is a mini Western. And then after that we rehydrate the gel. The printing takes 4 hours. Okay, because the array, each spot of the array is 300 picoliter. We print 20 drops. That means 6 nanoliter. The protein concentration needed is around 5 nanogram per nanoliter, or 5 milligram per ml. So we print 6 nanoliter per spot, and then we repeat the printing 10 times. The reason to print it 10 times is we want to let it to have enough time to go into the gel. If you print 60 nanoliter at one time, the spot will be too large. It will spread out, and then the 2 spots near side will touch each other. It will interfere your data analysis. So in order to avoid that, we print 10 times. Each time 6 nanoliter. So they will actually go a little bit into the gel. So after rehydrate, we do an electrophoresis on a GE genophobe. That means horizontal dry electrophoresis for only 12 minutes. So the separation will be 1 millimeter. Because the spine is so small, only 60 nanoliter compared to 20 microliter you load onto a regular Western. It's almost like 1,000-fold less. So 1 millimeter separation is enough to separate most of the protein. We have analyzed protein that has different larger than 10 kilodalton, we can tell on this gel. And then after that, we use a regular transfer box to transfer onto natural cellular membrane, and then put into a gasket, 96 well gasket. That has a rubber sheet that can separate all the different well. So we can put different antibody into each well. That means you can put maximum of 96 different antibody in a membrane. Okay, and so one pin can print 1 gel for 4 hours, and if you put 4 pins, you can actually print 4 gel at the same time within 4 hours. So that means if you use 4 pin, you can get 384 mini Western. And the liquid, the depth volume needed for the nano product is 25 microliter. So it's about like 1 or 2 times used for a regular Western. And like in here, we can do 384 times. So that's how the Gaussian nano product look like, not very big compared to the computer. And then that's the Lycon honestly infrared scanner used to scan, because it's no way you can see those spots on the beam. The only way you can see that is to observe them on the scanner. The scanner has a minimum resolution of 21 micron, and it's the infrared. So infrared scanner of the Lycon has some advantage compared to the traditional chemical way to amplify the signal. The infrared is actually linear. The chemical way is non-linear, but infrared is linear. And then the second thing is, for this scanner, there is 2 wavelengths, 680 and 780 nanometer. That means you can put 2 different antibodies in one wave. For example, like one rabbit and then one mouse, one for the 680 and one for the 780. So that means you can put like one for the EGFR, the other for the actin. So each well can have internal controls. So as the validation here, now we publish on the Nature method this January. The traditional Western look like this. We dilute the protein marker like 2 times each time. This is the image you see on the Microwestern. So it's like a circular spot. So the infrared dye is linear. The protein signal is linear compared to the protein you put. So that's very easy for your data analysis, because anything you observe, the change for is proportional to the protein change. And so we compare like different molecular weight of the protein marker, and all of them look linear. Then we look at different protein, gap gauge, phosphor, AV1, phosphor ERK, phosphor EGFR, phosphor Crackle, Curin, and phosphor AKT. We also do the dilution, and it also looks linear. So that proves the signal. Second, as the paper I just mentioned by the Marquez group, they say that the reverse-based array only 4 out of the 61 antibodies look identical with the traditional Western. That's why we want to validate if our result is identical to the traditional Western or not, because we don't want to waste time to do the traditional Western again after our Microwestern. Sometimes the sample is so limited, you don't have enough sample to repeat that. So we compare 11 different antibodies, phosphor EGFR, different tyrosine signs, and then phosphor IRS1, AKT phosphor, ERK, phosphor MEC, P90 ISK, phosphor AV1, phosphor Crackle, and gap gauge. The red signal is the antibody. The green signal is the actin. Okay, I already told you, you can put 2 antibodies in one well. Here is the traditional Western's image, and here is the Microwestern. 3 triplicate. And the red signal is the Microwestern, the macro, that means the traditional Western. The blue is the Microwestern. This is the sample of the EGF stimulation in the F431 cell, different time point. You can see the gray for 3 different experiments is almost identical to the traditional Western. So there is no difference between the Microwestern and then the traditional Western. So the cool thing here is anything you see on the Microwestern, you don't need to repeat that in your traditional Western.